So thanks a lot for allowing me to come and talk to you guys about my work. So just to get a rough idea, how many of you actually work in on DNA methylation or epigenetics? Okay, so some of you will be very familiar with the next few slides at least. So the basic structure of my talk is that I will go through, I'll give you an introduction, I'll tell you what DNA methylation is for those of you who are not very familiar, and I'll tell you about why we think it's important. And then I will give you a background about the data that we've generated. So I work a lot on whole genome data sets using bisulfite sequencing. So I'm going to tell you about how we generate that data and how we analyze it. And then I'll get into the most interesting part, which is what we find in these data sets. So cytosine methylation occurs primarily in the context of CpG dinucleotides, and it is mostly a symmetric modification, meaning that the C on one strand is usually modified on both strands if it's modified on one strand. We know that DNA methylation, well, we have clues that DNA methylation is important, especially in the context of development. And part of the reason why we believe this or know this is due to work that's been done on the enzymes that actually perform the methylation of DNA. And these are called DNA methyltransferases, and they come in two varieties. You have the maintenance methyltransferases that recognize hemimethylated DNA and are in charge of propagating DNA methylation through mitosis. So it's a heritable mark. And then you have de novo methyltransferases, and they're responsible for they act primarily on naked DNA and are responsible for providing methyl marks to cytosines, and usually they operate during development. And different methyltransferases are important for, they're expressed, we know in embryonic cell lines, in neural cells and hematopoietic cells, and experiments that have been done in these systems have shown that in some cases they're required for proper differentiation, and if they're mutated, then we know that there are anomalies that are associated with their dysfunction. So CpG suppression is a feature of mammalian genomes, and what this means is that CpGs are not randomly and evenly distributed throughout the genome. They're normally found in clusters. And actually, the reason for this has to do a lot with the fact that CpGs themselves are very infrequent in a mammalian genome, in a typical mammalian genome. And part of the reason for this is that over evolutionary timescales, we've experienced a loss in CpG sites. And this has to do with the fact that methylation itself undergoes a biochemical change, so it gets deaminated to thymine instead of uracil, whereas unmethylated cytosines normally would be converted to uracil under deaminating conditions. And depending on which, if it's methylated and deaminated to T, or if it's unmethylated and deaminated to C, then it's better recognized by the mismatch repair machinery. And C to T transitions aren't very well recognized, and therefore they get transferred to the next generation as a mismatch, as a T transition. So over evolutionary time, we've experienced a loss in CpG sites, and this has created CpG. This is what's actually created CpG islands. So you can see here in this illustration, we have dispersed CpG sites not located in close proximity to one another that are methylated, whereas we have here a very dense region of CpG sites that are unmethylated, and they get sort of retained throughout evolution. So methylation in mammals is mostly erased during gametogenesis and pre-implantation. And it's replicated through mitosis and some evidence that it passes through meiosis. But what are the actual functions of DNA methylation? And I think this is something that has really come to the forefront and is part of a larger debate in genomic science. But what we think of when we think of methylation is that it's associated with transcriptional silencing. And the reason why we believe this is because when we see methylation at promoters, which are mostly unmethylated, you have a correlated silencing of the expression of that promoter. So we believe that it does have something to do with gene regulation. And we also believe that DNA methylation, there are at least some evidence in the literature that DNA methylation is important for guiding cell fate specification. And the way in which DNA methylation possibly interferes with gene expression or gene regulation is by blocking transcription factor binding. And there's also believed to be a relationship between chromatin modifications and DNA methylation. But the cause and effect relationship is still actually being investigated, and trying to understand which is important at which step is still under debate. So in order to investigate genome-wide DNA methylation, we require several things. And we need a method that will allow us to look at single base resolution, or at least single CpG site resolution. And we also need a method that will be agnostic actually to sequence context. So we don't want something that's only going to look at CpG islands. And we also want something that's going to be robust to or very sensitive to finding levels of methylation at specific G sites, the CpG sites. So we want something that can actually identify the specific frequency of methylated cytosines at this single CpG site. So the method that we've chosen, and probably the most frequently used method for this kind of requirement, is bisulfite sequencing. And we're actually using the Illumina platform for doing whole genome bisulfite sequencing. So we're doing shotgun bisulfite sequencing. And what this means is basically that methylation is a very stable mark, and it can be found on intact. It's found on intact DNA, and doesn't require intact chromatin to actually to find the location of these marks. So we use bisulfite sequencing, and bisulfite basically means that we're using sodium bisulfite to convert unmethylated cytosines to uracil. So what happens is you expose the DNA to sodium bisulfite, and then this deaminates unmethylated cytosines to uracil, and then uracil then gets converted to T in the nascent strand when you actually amplify the DNA. Methylated cytosines are protected from this conversion. So what I'm showing you here is that you have the top and the bottom strand, and you have methylated Cs and unmethylated Cs. You have both cases. You denature the DNA, and then bisulfite converts. So then you actually have two strands that become converted, and they're no longer able to anneal to one another because they're no longer complementary. And then the deaminated Cs get converted to T when you do the PCR amplification, and then you have actually 4 strands, whereas before 4 unique strands, as before you had 2. Then we make Illumina libraries. So what happens is we fragment the DNA. We ligate on adapters that are methylated, and so the adapters actually have Cs in them, so we have to keep them from getting converted, so they'll actually still be compatible with the sequencing technology. And so we ligate adapters onto the DNA, and then we do bisulfite conversion. And then we do high throughput sequencing, and we require at least 10x coverage. So that means we need at least 10 reads per base in order to call methylation. But first, we have to actually map the reads back to the reference genome. So for all of the...
Why does Bisulfite not convert methylated cytosines into time?
Without knowing the actual chemical, structure, the chemical process, somehow the modification protects the cytosine so it doesn't get converted. I don't actually know the hardcore chemistry behind it. Sorry?
You expect it, right? The deaminated, you would expect. If it's deamination, you expect it to convert to time.
Yeah, you would.
Somehow it's protected.
In this case, yeah. So I don't know. I don't know how in this case it's protected because it's a different. I guess it's a different. It's a different. Right. Okay. Can you say that again? Right, but I guess the question is why. In the case of what I was explaining earlier, how methylated cytosines can be converted to Ts over evolutionary timescales. Why in that case it's a deamination process, and why in the case of sodium bisulfite, the chemical reaction that we use to convert unmethylated cytosines, why they don't get converted to Ts in this case. And I'm actually not sure. But it's an interesting question. Right, so for all of the work that I've done, I've collaborated very closely with a wonderful computational biologist, Andrew Smith. He's a PI at the University of Southern California. And he's developed a lot of tools for mapping bisulfite sequencing data, and also a lot of analysis tools for doing many things that I will go into a lot more detail about in a few more slides. But just to explain the problem with mapping bisulfite reads, the biggest problem is that you actually reduce when you subject the genome to this conversion process, you're actually reducing the complexity of the genome in the sense that you're essentially reducing a 4-base genome to a 3 or a 3.5 base genome. So this creates a problem in the sense that it creates bigger ambiguity when you want to map the reads back to their reference location. So Andrew has done a lot of work creating a software, developing a software called RMAP, and he's implemented specific modifications to this RMAP software that it allows you to actually map bisulfite reads back to the intact reference genome. There's several different methods out there for doing this. We can certainly discuss them, but at least in the case of RMAP, there are specific rules in terms of mapping. First of all, the T's and the reads T's can match a C in the reference, but only if a T is followed by a G in the reads. And there are several different other modifications that he's added to the software in order to combat the problems with reduced sequence complexity and actually dealing with ambiguity and mapping that can occur. So then we need to understand how we're going to call methylation. So methylation is great because it can give you a sense of what the population of cells actually looks like, because when you're mapping methylation levels or methylation frequencies at a specific CpG site, you're actually looking at individual molecules that that pile up on the sequence, and each individual molecule maybe represents an individual clone of your tissue of interest. And so this gives you a sense of the heterogeneity of the population. So normally we see methylation levels approaching 0 or approaching 1. So if it's a fraction of Cs that are actually methylated at a given site. So you can have different types of methylation. We have where we see, so I didn't explain that very well, but the idea is that, yeah, this is better. If your reads pile up on the reference genome and you have different reads representing a CpG site, then you're actually counting the frequency of times that you see Cs versus the frequency of time you see T over the number of reads that you're actually looking at. So that will give you a frequency of CpG methylation at that specific site. I'm sorry. So we have different types of calls that we can make based on these frequencies. Either we call it unmethylated, and so if we see that below 25%, for example, of Cs are methylated or unmethylated at a given site, then this is unmethylated site. If it's more than 75%, so more than 75% of Cs are present at the CpG site, then it's methylated. So in the sense of the unmethylated, if it's Ts at the site and less than 25% Cs at the site, mostly Ts at the site, then it's unmethylated, if that makes sense. And then we can call something as partially methylated. If we have enough reads covering the site, we can statistically confidently call partial methylation, where you have maybe 50 and 50%. You'll never have, it's very rare to have that unless it's allelically methylated, but in some cases you do have partial methylation where you see 50% Cs and 50% Ts at a given site. So this is just again showing you example of reads covering these CpG sites. So they're aligned to the genome, and the alignment and the frequency of Cs to Ts gives you an idea of the methylation level at those sites. So how many of you are familiar with the UCSC genome browser? I'm going to be showing you guys a lot of genome browser shots, but I honestly think it's one of the best ways to visualize the data, yes?
You said that you use a coverage cut of 10. Is that it has to be 10 methylated and 10 unmethylated, or an overall coverage?
10 in whole. I mean, obviously the more the better, but we actually have to have at least 10x coverage for the entire genome. And I mean, just to give you an idea, before the HiSeq, we were doing this on GA2s. So now that the Illumina platform has been upgraded, so you can generate 250 million paired in 100 reads in a single lane. But before it was more like 4 times less, and so we were having to use an entire flow cell just to get 10x coverage, and that's actually not as much as you would need to map an unconverted genome at 10x coverage. We need a little bit higher sequencing capacity to do it with methylated DNA. With bisulfite sequencing. Yeah, so, 10x is a minimum cutoff, and it's also minimum and most cost-effective cutoff for doing this genome-wide. Now it's getting better. Now we can do on an Illumina HiSeq, we can do 1 to 2 lanes to get that amount of coverage. So if you're looking at the genome browser wiggle track of the data, this is we're going to be showing you a lot of these. On this axis, we have methylation frequency, and each bar represents a CpG site. And this is a zoomed out view. So you might have, for this 100 kb region, a number of genes, and you can see at each transcription start site for every gene, these dips. And those regions are unmethylated. So This is just a zoomed in view. And a view of a single gene, DNMT1. There's a CpG island located at this promoter. And you can see that this region is highly hypomethylated, whereas everything else is methylated. And this is the methylation, what we refer to as the methylation pattern. And this actually brings up another very key point. So in most mammalian methylomes, 70 to 80% of the CpG sites are methylated. But we were interested, so this sort of brought up the point that we didn't want to actually look. One of the reasons why we wanted to have an impartial method for profiling methylation is because we didn't want to actually look at a CpG island-centric view. We wanted to be able to identify regions of interest without focusing in on specifically CpG islands. And I hope that the reasons behind this become a little bit more clear. But the reason is that 70 to 80% of CpG sites are methylated. So this means that what we should be looking at as let's say a biological signal of interest is actually hypomethylation. And this way, we believe that we might be able to search for regions of regulatory importance in an unbiased manner. Yes?
I was about to ask if that's like 70 to 80% of CpG islands are very localized novel promoters or no HMRs.
Right. So what you bring up is, so actually probably more, but at least we know that at least 70% of promoters are hypomethylated, but probably more. At least 70% of promoters are associated with an annotated CpG island. And we know, so the CpG islands that are located at promoters are they're part of what we call these high density CpG islands. A lot of people have tried to categorize CpG island into sort of lower, middle, and higher CpG density, and the most highly CpG dense regions tend to be constitutively hypomethylated. So no matter what cell type, no matter what developmental stage, they're always going to be hypomethylated. And those are the ones that are mostly associated with promoters. And but there are changes there, and I'll talk about that a little bit more as we get into it.
It's a nice picture that shows low methylation on the shores of the CpG island. Would you think that, I'm just wondering about the definition of a CpG island and whether that definition should be maybe changed or extended. You know, it seems that maybe a CpG island should actually be a larger version of what we call CpG islands now.
Yeah.
Is that the case or not?
I completely agree. I don't know if, I mean, CpG Island has been, so I guess it's more of a historical problem, because it's been defined in a specific way, and we use specific criteria. The CpG Islands that are defined, for example, in the UCSC genome browser, are defined based on a specific set of criteria. They must be above a certain observed over expected rate for CpGs. They must be above a certain GC content. They must be bigger than a certain size. And I'll show you a little bit later how the regions that we identify as hypomethylated, we find many ones outside of the criteria that define CpG islands. So ones that are even newly hypomethylated, so not associated with an annotated CpG island. And it is true that the borders of the regions actually fluctuate, and they fluctuate especially at promoters. And I'll show you a lot of examples of that. In terms of defining what a CpG island is, I think it's very difficult. I think that's why this kind of data is extremely crucial, because we're not actually biased towards any set of criteria to define based on sequence. I mean, I think that those definitions are very important to give you sort of an initial view of the genome, but in terms of actually finding all of the methylation important regions, then it's very difficult just to focus on one set of criteria. So, but I agree with you that CpG island is no longer a useful term. Yes.
So aside from promoters, are there any other genomic features that CpG islands are consistently associated with, like enhancers or something?
Yes, I mean a lot. So this is part of the issue. So in our data, we find a lot of intergenic hypomethylation, and sometimes sometimes it's associated with the CpG island, and sometimes an annotated CpG island, just to be clear, and sometimes it's not. But we do find these clusters of hypomethylated regions that we believe that do overlap features like histone marks that are associated with regulatory elements. And so there are CpG islands that have been annotated outside of promoters, but part of the problem has to do with the fact that the sequence criteria doesn't take into account genomic context. And in intergenic space, the sequence characteristics are are quite different than you would find sort of in genic regions. And so there are many, what we might refer to as low CpG density CpG islands, but there's still methylation, they're still significant in the context of changes that might happen in methylation. So as I mentioned, we're interested in hypomethylation as a signal of interest. So Andrew has worked very hard to define a hidden Markov model that will identify HMRs. And if you're interested, we can certainly go into more specific detail about this model. But basically, it allows you to identify these regions so it's sort of an unbiased strategy for identifying these regions. And it's devised to detect the clustering of these unmethylated CpG sites. As you can see, these bars, and the bars that I will show you throughout the rest of the talk are HMRs that have been defined by this model that Andrew wrote. Okay, so we've generated bisulfite sequencing data from many different cell types, and we've also taken advantage of publicly available data sets that pre-existed and then are continually populating the public databases. But what we find is that in many cases, HMRs, and this is a hypomethylated region, just to be clear, an HMR, as we have defined it through this modeling of our bisulfite sequencing data, HMRs are conserved in many cell types. So all of these cells, these are all blood cells, and they're blood cells that I have generated here. This is this was another data set generated by another lab. But you can see that there's really high conservation between HMR calls, and the boundaries are very defined. And then if you look at cell types that are unrelated to blood, so from totally different lineage, you see that there are other HMRs that come up that are more specific to lung, for example. So there's constitutive HMRs that are found in every cell type, and they're usually associated with the TSS, as I mentioned. But then there are more cell type specific HMRs that are found a lot of the time in intergenic space associated with a specific lineage. So as I mentioned before, differential methylation and expression. I think this is one of the few cases in which you might see methylation associated, like the methylation state of the TSS associated with differential expression. So this gene is CD19. It's a cell surface marker for B lymphocytes. And so it's specifically expressed in B lymphocytes. And this is this HMR is found at the TSS of CD19, and it's not found anywhere else in any of these other cell types, including the blood cell types. So this is one of the few examples where you see a specific HMR located at TSS that's associated with higher expression. Most of the time, an HMR is shared, as I mentioned before on the previous slide. Most of the time, HMRs are found at every single promoter, and at every single promoter in every single cell type, so that's constitutively hypomethylated. And so any change in methylation, it's very difficult to correlate methylation in that case with specific changes in transcription if they're always going to be hypomethylated at the promoter.
Does that really correlate with just transcription instead of differential transcription?
Right, no, it doesn't. I mean, because so there are very subtle changes that happen, but most of the time there's a core region that's always hypomethylated right around the TSS. And I think it'll be a lot clearer maybe in this example.
Do you have any idea about what's so special about this gene?
Well, it could be that the most cell type specific, the ones that are sort of ubiquitously expressed or modulated in very subtle ways, may be constitutively hypomethylated, and then very subtle changes that occur are associated with these subtle changes in expression. But maybe the ones that are really, really highly tissue specific, maybe that's one difference. This gene, though, is another example. This gene is it's also a cell surface marker, and it's specifically upregulated in B cells. And you can see that in most of the B cell, there is a region that's constitutively hypomethylated. There's two regions actually. And none of these regions are actually hypomethylated in any of the other cell types that we've looked at, including sperm, lung cells, embryonic stem cells, or fat cells. But what's interesting about this gene is that it's hypomethylated specifically at the TSS, and then suddenly it spreads. So this gets back to the shore thing you mentioned. So we do notice that the boundaries of HMRs actually change, and one of the ways in which they change is by expanding. And mostly, when we see this HMR expansion at a gene, we see the expansion occurring in the geneward direction, and this is actually associated with higher gene expression.
So I have a question about causality here, because to some extent, transcription also changes epigenetic state, histomodifications, and those are coupled with methyltransferases.
Yes.
And methylation, right?
Absolutely.
So it could be that transcription downstream also changes methylation state. It's hard to separate it from upstream and downstream of it. So could it be that the second through fourth line correspond to past polymerase states, and then it's going to...
I actually believe that this is, I think that this is probably a very good theory. We have, so if you actually look at the histone pattern at these types of regions, you see the spreading of histone, and I have some inkling that it could be related to the activity of polymerase. I don't know that anyone specifically testing this or has tested this, but it's difficult to say what the cause and effect is. But we have some idea that the action of polymerase could cause that. I think it's very fair to say, and very interesting too. So another thing that we think is interesting about finding HMRs is that we can use HMRs to find specific types of regulatory elements. We know that transcription factor binding sites, for example, are enriched within the HMRs that we've identified. How are we doing on time? Okay, good. And another thing is we've done, so another interesting thing that we've done is I'm very interested in doing species-specific comparisons. So we've generated for every blood cell type, in addition to sperm, we generated equivalent bisulfite sequencing data sets in chimpanzee, and we've compared them to each other, and we find that they're highly, highly similar. So we've used, we've taken chimp reads and lifted them over to the human genome, so that's why you're able to see the data like this. And this is a specific example of a bidirectional transcription start site that's shared. The patterns of methylation are shared between chimp and human. But then we have other examples of genes that are potentially differentially regulated, and we can identify these regions through bisulfite sequencing. So this is a great example of a gene where in all of the blood cell types, you have one isoform that's hypomethylated, two isoforms that are hypomethylated, whereas in the chimp data set, only one isoform is hypomethylated. And I just thought that was kind of a cool example. Another thing is, aside from genes, we can also look at transposable elements. Transposable elements are normally suppressed by methylation, and so we can actually find transposable elements that have transcription start sites and are unmethylated at their transcription start sites. So there's a lot of different types of ways in which we can use this type of data to identify regulatory features and genomic features. So for the rest of the talk, this was sort of an overview of the type of data that we've generated. This is an overview of how we've analyzed it and the types of things we're looking at. I wanted you to be able to visualize really what we're trying to do and what's interesting about it. And there are a few key points that I think you should sort of take with you as we go into the next parts of the talk. And that is that, we have several assumptions that we can make. First of all, methylated DNA may not be accessible for TF binding, and this may be one way in which methylation sort of suppresses the regulatory element. Unmethylated promoters are usually open for business, and this means that they can be open for the assembly of the transcriptional machinery, pause polymerase, or they're open for expression. And DNA methylation data can be used to understand whether or not there's a presence or an absence of an enhancer, for example. And I'm going to show you a lot of data attesting to that idea. And it can also provide information about the heterogeneity of a cell population. So, for example, if I found a region that was 50% hypomethylated, and the indications are that this methylation is not an allelic pattern, then it would indicate that the population of cells actually has potentially a heterogeneous phenotype. But most importantly, I think, is that hypomethylation can help to identify the precise location of interesting and important regulatory features.
So, for the first point, if you say methylated DNA is accessible for TF binding, do you think of that at the regional scale, say of the chromatin domain, like a methyl binding protein recruiting an HDAC and then shutting down a chromatin? Is that kind of your picture there? Or?
Yes, I think that, without knowing the precise mechanism by which, it's again a question of cause and effect. So we know that in some cases, so this is actually, there's no rule for transcription factor binding. Some transcription factors have been shown to be, to have a lower affinity for methylated DNA. So they don't bind DNA that's methylated. But then others can bind methylated DNA and cause demethylation of the regions. The stable binding of the transcription factor is dependent on the region being free of nucleosomes and free of methylation. But again, there is a model for transcription factor binding actually causing demethylation. But I think for the stable binding of a transcription factor, it needs to be unmethylated. It's just a question of what comes first. Yes. So now I will talk a bit about how we've been using this kind of data and the kinds of questions that we've been interested in. So we're very interested in how DNA methylation, what kind of role DNA methylation may play in cell fate specification or differentiation. And so we set out to understand how the DNA methylation profiles differ in different cell types and at different stages of development. And for this, we used the hematopoietic system because it's a very well characterized system. The cell surface markers used to purify cell types have been very well characterized, and so that was actually an important requirement for us, that we be able to isolate specific cell types at specific stages of differentiation, and that we be able to do this very pure. So we actually started out using 3 different cell types. We used hematopoietic stem and progenitor cells, and these cells are the cells that are capable of self-renewal, and they're capable of giving rise to all of the different cell types in the blood lineage. And there are two blood lineages. There's the myeloid lineage and the lymphoid lineage. So we actually wanted to have 2 cell types that were representative of each lineage. So we have sort of a triad of cell types that we looked at. We looked at the stem and progenitor cells, and then we looked at neutrophils, which are part of the myeloid lineage, and lymphocytes, B lymphocytes, which are part of the lymphoid lineage. So we were able to sort of reconstruct the changes that occur between the hematopoietic stem cell and sort of differentiation to the terminally differentiated lymphoid cell, and the terminal differentiation of the myeloid cell, and we were able to look at specific changes that are shared with the stem cell and then not shared in the differentiated cell types, either to each other or to the stem cell. And we were also able to use other data sets as an outside comparison. So we were able to use the embryonic stem cells that were generated by Joe Ecker's group, Ryan Lister and Joe Ecker's group, back in 2009, and also germ cell data sets that we generated in the lab.
What species were you studying?
Human. This is all human.
And you're defining the HSCs as CD34 positive?
CD34 positive, yes. Lineage negative, lineage marker negative, and CD34 positive. So that's why I'm kind of I'm calling them stem and progenitor, because it's not a pure population of long-term self-renewing HSCs. We isolated them from peripheral blood. Yeah.
After stimulation or
Just no stimulation. And this was tough because we didn't get very many, and so we had to generate methods for doing this on very low input number of cells. Yeah. This is just a reiteration of what I mentioned earlier. Here I'm looking at a myeloid-specific region. So this is a myeloid gene and a CpG island that overlaps these 2 promoters, these 2 transcription start sites. You can see that this CpG island is constitutively hypomethylated in all of the cell types, but the boundaries fluctuate a lot. They fluctuate more than in the other regional examples that I showed you. And then there are here out in the distance specific HMRs that are only found sort of in the blood cell types. This is an example of a region where it's out in the middle of the genome. There aren't any close genes close by, and it's a cluster of HMRs that are found only in B cells. So it's something we'll talk about a little bit more later. So if we take the genome coordinates of all of the HMRs and we try to overlap them to see where they're shared and which ones are cell type specific, we find that within at least blood, many of the HMRs are shared to some degree between all of the blood cell types. But then you find, thousands of regions that are actually cell type specific. If we look at how a blood cell overlaps a sperm cell, for example, you see that there are many, outside of the lineage, the tissue-specific lineage, you see many more tissue-specific HMRs. And just to give you an idea of how these HMRs overlap predicted or annotated CpG islands, basically that's what I'm showing you here. Most of the HMRs that we call overlap the annotated CpG islands, but look how many don't. There's just so many more that actually don't overlap an annotated CpG island. And this is one thing that we found very remarkable. So it means that you can't use sequence-based criteria to define CpG islands. And this is just a clustering of the HMRs in different from the different cell types. You can see that the blood cell types cluster very closely together, and the other cell types, as you would expect, are farther outside. Okay, so as I mentioned, there are specific criteria for calling CpG islands. And the cutoffs are that they have to be over 200 base pairs, they have to be over 50% GC, and they have to have an observed over expected rate of 0.6. And that's actually really high, because the frequency of CpG nucleotides in the genome is probably 4 to 5 times lower than any dinucleotide with a G or a C in it. So it actually is a very stringent cutoff for a CpG island, I think. But anyway, I'm showing you basically the different quadrants at 0.5% GC, 0.6 observed over expected, and how all of the HMRs sort of fall into these different quadrants. And you can see that these would be the equivalent of CpG islands. And there are so many more that don't sort of fit this criteria. If we look at the context of the HMRs that we call, we find that many are genic. Actually, many of them do overlap CpG islands, promoters, sorry. But there's actually a lot that are found outside of genes. And that's, I think, a critical point that we didn't necessarily expect. If we look at differentially methylated regions, that's what I mean by DMR. So if we compare the different data sets and we look for where the differentially methylated regions are, we find that most of them are intronic or intergenic. So this means that the HMRs that are cell type specific, the ones that are most crucial or potentially crucial for a cell type identity, are located outside of genes and actually in intergenic space and also an intronic space, and are actually associated with the promoter. So this was actually the example I showed you. It's CD19 in B cells. You see that there's an HMR here, and where you find the highly dense CpG island, you see that there's constitutive hypomethylation here. And this was the other example that I showed you, CD22. So you see there's shared hypomethylation at the TSS in different blood cell types, but an expansion of the HMR in B cells, and this is correlated with, maybe kind of difficult to see here, but I've also overlaid a track that shows RNA-seq reads. And there are many more RNA-seq reads across this gene in B cells than the other cell types.
It does seem like the non-CpG island HMRs are not as hypomethylated as the CpG HMRs.
That may be true to a certain degree, and certainly you see in the expanded part that there's sort of this ramp. That in the case of the shared of the shared HMRs, the boundaries are much better defined. The borders of the HMR are better defined than the ones where you see this sort of fluctuating methylation levels, CpGs. Right. So we asked whether or not this is sort of a global phenomenon, whether this is actually happening, because if you have a constitutively hypomethylated or shared HMR at a TSS for all cell types, then how do you infer what the expression state of that gene is? Because you can't really infer that with confidence that it's actually being expressed. So we wanted to see if the expanding promoter idea was something that could be applied globally. So if we look at aggregate plots over the TSS of all genes that are sort of either hypomethylated in B cells or hypomethylated in neutrophils, and we look at the methylation level in both. And this is sort of difficult to see, but you can see that on the GeneWord side of the TSS, there's this gap. There's sort of an expansion in the hypomethylation. And this is, if we identify differentially methylated regions, so this is a separate identification than identifying HMRs. We're actually looking for places where we actually see significant differential methylation. And we plot DMRs across the region. We see that there's an aggregation of DMRs on the GeneWord side of the TSS. So that sort of supports what we observed in the first place. And then if we look at the correlation between differential methylation and expression, we see that this is actually, I think it's probably better just to look at it here. Methylation versus RPKM, the axis is inverted so that you see a peak rather than a valley, but because methylation and expression should be anti-correlated. And this is basically what you see. We see that the correlation or the anti-correlation is higher where we see differential methylation or where we see this expansion of hypomethylation at the TSS. And these are aggregate plots of histone marks at these genes. So we took advantage of ENCODE data sets that include lymphoblastoid cell lines. So we used the lymphoblastoid cell line as a proxy for our B cell primary lymphocytes. And we had a lot of histone data at our disposal from ENCODE. And so we were able to look at aggregate plots of histone methylation. And what we find is that indeed the histone patterns actually match the expanding HMRs. And I think I have a much better plot that I can show you a little bit later. But what I'm basically, the points that I'm trying to sort of drive home are that there's a genic bias that emulates the spreading of active histone marks where you have hypomethylation at TSSs. And this may be important for the fine-tuning of gene expression upon differentiation. And it is consistent with the concept of CpG Island shores that was originally described by Andy Feinberg. So let's see. I just want to check where we are. Don't get scared. We're almost finished. Okay, this is almost the end of this part. Okay, so one thing I mentioned before is that DMRs are located primarily in intergenic regions and in intronic regions. So we were interested in understanding what these HMRs and intergenic regions could possibly be, with the hypothesis that they represent potential regulatory elements. So the first thing we asked was whether or not HMRs show any evidence for conservation, because often signatures of conservation are used to identify regulatory elements. And comparing these regions to promoters, for example, we see that methylation level, first of all, intergenic HMRs are a bit shorter than promoter HMRs, and their methylation levels are a bit higher. But they're also conserved. The conservation scores are a bit lower, but they still have pretty significant conservation scores compared to flanking regions. So we believe it's fair to say that these HMRs harbor regulatory elements of some kind. And we found that these regions are also enriched for motifs for transcription factor binding site motifs. And the enrichment is correlates very well. So here you're looking at regions that are myeloid specific, so neutrophils are less methylated than B cells, or in this case, B cell specific, where B cells are less methylated than neutrophils. And in each case, you see that there's very cell type specific transcription factor motifs enriched in the neutrophil specific case and the B cell specific case that supports, yes? Yes, absolutely. So we define intergenic as anything greater than 2 kb away from a transcription start site. Okay. Or, greater than 2 kb away from a gene. Yeah. And not overlapping any kind of non-coding RNA or we actually had to, in this data set, we also excluded HMRs that overlap repeats as well. So that actually gets rid of a lot.
Mm-hmm. And yeah. Like ESPs, you consider it as gene or
no. So no, we don't. Unless usually we use RefSeq as the manually curated RefSeq data set as the gene data set that we define for whether or not the HMR is overlapping the gene or not. We use RefSeq to find genes.
So in this way, it could also be that it's just a TSS that is more upstream than what is embedded in RefSeq.
Yeah, I mean, in principle, it's possible. It is possible. I mean, it's possible, I guess. That's an alternate promoter of some kind. But most of these regions are located like greater than 10 kb away from the gene. So, I mean, it's possible, and it's also possible that they're the TSS of some non-coding RNA or link RNA, which is something I've seen a lot actually. Yeah. But the point is, I guess the most important point is that they're the most cell type specific of all of the HMRs that we actually find, and this is probably the most crucial thing. They're the ones that, regardless of what kind of, and it's actually something I will go into after the break, but they're the ones that are never found anywhere else except for that cell type, and I think that's regardless of whatever type of regulatory element or gene structure that may be there, they're always most cell type specific.
So these are from JASPAR or something, that they're predefined?
Actually, I think we used, in this case, I think we used the TransFact database.
Okay.
Yeah.
But they're predefined.
Right. Yes.
Discovering those in your data.
No, not. And then the last few slides, I just want to sort of giving you a little bit of a teaser for what I'm going to talk about after the break. So if we're just specifically focused on looking more into analyzing the intergenic HMRs, we find that there are some intergenic, I did say that most of them are very cell type specific, but there are some intergenic HMRs that are shared between cell types. And we found that they're highly enriched for CTCF, so a genome organizing insulator protein that's often found to bind these shared intergenic HMRs. Another thing that's interesting is that, so this is ChIP-seq data sets from POL2 and PU1. It's actually a B-cell specific transcription factor. And what you can see is that this intergenic cluster of HMRs, you see more hypomethylation in B cells, and there's some evidence for, and this is actually coinciding with where this transcription factor is binding. And we also see POL2 activity here, which indicates that there's actually something going on at this region, but we're not sure. Not sure exactly what it is. So the last slides, we find that intergenic HMRs can actually be transcribed. So this is a B-cell specific cluster. I'm basically superimposing on these tracks H3K histone marks, so ChIP-seq data from ENCODE, again from lymphoblastoid cell lines, looking at H3K4 monomethyl and H3K27, acetylated H3K27, both marks associated with active enhancers. We see that there's a very strong overlap between these sites and DNA clusters that have been identified in this cell type, and transcription factors that bind to these regions. And we also find that there's some evidence for RNA polymerase activity. Although the RNA-seq data sets it's very difficult. Most RNA-seq data sets have very low abundance reads in these types of regions, so it's difficult to look at most of the existing RNA-seq data sets, but we do see that there's something there.
This looks almost like one of those super enhancers that have been described recently.
Oh, I'd love to talk about this. So, I mean, I hope we can talk about that after. I think that it was really interesting because when I saw the papers, I saw him speak at a recent symposium at Cold Spring Harbor, and it was truly very interesting. But what fascinated me is that he actually defined this new thing, the super enhancer, but something we've seen in our data a lot, and it's something I'm sure others have seen, where you have clusters of, I mean, you can see that the regions are rather small. I mean, they're an average intergenic HMR might be like 500 base pairs, 300 to 500 base pairs or less, and a promoter HMR, by contrast, is 1 to 2 kb. So they're smaller, but they're often found in clusters. So you can find a single HMR that resembles an enhancer out along by itself somewhere, but very frequently they're located in clusters together. And I think maybe they could be called super enhancers. Who knows? Yes, but you're absolutely right. So this brings up a lot of questions that we'd like to ask. Do IHMRs harbor non-coding RNAs? Are they pervasively transcribed? A lot of people who work on these kinds of data sets, work on ENCODE, would say yes, that transcription is pervasive transcription is a feature of the genome. Are IHMRs enhancers? How do IHMRs actually interact with PHMRs? So promoter HMRs is what a PHMR is. I'm going to use that. I'm going to use the HMR term a lot, so sorry. Yeah, so how are our IHMRs actually involved in gene regulation? And these are all things that I will talk about after. the break. So, thank you. Any questions? Any more questions?
Yeah.
Yes, you go first. We'll go around the room like that.
Did you see any correlation between the transcription of the EPUB servers with the transcription of your actual genome?
Yes, and I'll show you some more. I'll talk a lot more about that in the next part. Actually, it's going to be focused entirely on the next part. I'm going to focus entirely on the chromatin features of these IHMRs, the transcriptional features, and how they relate to activity at the actual nearby gene. Yeah, I'll talk about that a lot.
So there have been attempts to define kind of empirically with non-methyl binding proteins,
Mm-hmm.
like CSHL and things like that. Do
Right.
those correlate better with HMRs than the in silico UCSC method?
I haven't directly compared. My first instinct is to say that probably. I mean, I guess the method is only as robust as the ability to consistently repeat it, and the sensitivity of the method depends a lot on the sensitivity of, for example, the sequencing platform. But I think that methods like that would correlate very well with the HMRs. But I don't know if they have the breadth to see everything. Right? I'm sure that methyl binding domain ChIP-seq, for example, would at least very well correlate with a subset of the HMRs. Whether or not it would be able to identify everything, I don't know.
Did you say that your intergenic HMRs, for your cutoff criteria, you threw out all the ones that overlapped with repeats, or that were anywhere near repeat elements?
Yeah, so we were actually very careful about defining yes and no. So, I mean, where we're interested in certain things like motif enrichment and that sort of thing, we actually excluded repeat elements, or we segregated them into ones that overlap and ones that do not overlap repeats, because there's a lot of confounding effects of the repeat biology. Although, I mean, repeats can actually impart regulatory information, so that's important too. The next part that I'm going to talk about reanalyzed all of the data, and we never excluded any repeat. Like we keep the repeats. Yeah. Any other questions?
So in your view, the HMRs are reasonably large regions, right? I guess you would call, I don't know, think about your 10 nucleotides, 20 nucleotides, that would not qualify as an HMR. But do you still see any regions where it's more, where you see a statistically significant change in methylation?
Yes. They may not be called by the model for calling HMRs, but yes, we do see statistically significant differences at individual sites and even small regions of a few sites.
But I mean, if you have a hypomethylated about those, is it the same mechanism or is it something totally different?
Same mechanism. Don't know. It's possible. It could be. Well, the thing is, if we were doing single cell sequencing, it might be a one-off event. But I mean, if there's statistically significant differences, I mean, these are millions of cells that were, or in some cases thousands of cells that we're sampling that show this kind of methylation pattern. It's hard to say. I don't know what it means. Maybe there's really small, it's possible that there's very small like binding events that occur and that are important. I don't know.
Okay, well, thanks.
right, so now we're going to get to a story that's kind of closest to my heart, I guess. It's what I've been working on in the last year or so, and something I'm very interested in. It's a downstream analysis of the data sets that we and others have produced, and also integrating a lot of publicly available ChIP-seq and all kinds of chromatin-based data sets that are available through public consortia like ENCODE Project and others. So we've used the data that we've produced and also many other large data sets to, in an integrated way, to derive some kind of meaning from the methylation data that we have. So yeah, I think the title basically describes, I have very few slides in this part. I don't think I'll take the whole hour to talk about them, so please, this is the time to discuss and have any questions. I'm really happy if you interrupt and interject. Please do. So the title very well encapsulates what I'm going to be talking about, and I'm going to be talking about de novo demethylation and non-coding transcription at these intergenic HMRs that I described in depth earlier. I'll be going into a lot more detail about this. All the work that I'm going to describe has been a collaborative effort with a graduate student, Felix Schlesinger, who was a graduate student in Tom Gingeris' lab here at Cold Spring Harbor. He's a very talented computational biologist, and he's now working at Illumina in their computational biology department. So all of the data and work that I will describe is very much his. So as I mentioned earlier, intergenic HMRs are very cell type specific. And the example comparisons that I'm going to be going through over the next few slides are going to be a comparative analysis of H1 embryonic stem cell methylation profiles to terminally differentiated B lymphocyte methylation profiles. So the same B lymphocyte methylation profiles that I spoke about earlier in comparison to H1 data that has been produced by other groups. And these plots are basically showing you the number of HMRs we identified in each data set, and then categorized based on their genomic location. So whether or not they're intergenic or intronic, overlapping promoters, or we actually distinguish HMRs that are overlapping CTCF because their behavior is very dominated by CTCF binding, so they're a little bit on their own. But what you can see immediately, we have distinguished 3 different categories within these genomic contexts. Either cell type specific, meaning these are the HMRs that are only found in that cell type, or expanding, which means, as I described earlier, they're shared, but in one cell type they expand and not the other. So they have a sort of shared core region, and then in one cell type they get expanded. And you can see that the expanding category really is very unique to the promoter category of HMRs. So there's a lot of shared, very few extremely specific promoter HMRs, but a lot of ones that get expanded in one cell type versus the other. But the most striking thing that you will see here is that compared to the embryonic stem cells, the differentiated cells have the most cell type specific HMRs and many more HMRs found in intergenic space compared to H1 cells. And as I say here, the cell type specific HMRs in stem cells is actually 4 times lower than what we find in B cells. And the loss of methylation at these sites may be linked to cell fate specification and differentiation. That was the topic that I began with. Is the one that I'm interested in understanding how methylation plays a role in this process. So, as I mentioned earlier, also we hypothesized that HMRs, that just identifying HMRs alone can be one method for identifying regulatory elements. One of the more popular ways to do this, and certainly more prolific ways, aside from just using from sequence-based features alone, like conservation signatures and other things, one of the other experimental ways to identify regulatory elements is by looking at DNA's hypersensitivity. And so we wanted to know how well our HMRs overlap DHS sites. And what we find is that for the most part, HMRs they overlap very well predicted, or not predicted, but identified DHS sites. And for the ones that don't seem to overlap DHS sites, if you look at the DHS signal, and that's what this heat map is here, we're looking across H1 HMRs and the DHS signal. You can see that for most of them, there is some DNase hypersensitivity, except that for a subset of them, the DHS signal is slightly lower. But if you take the reverse and you ask how many DHS sites are hypomethylated, so how many DHS sites overlap an HMR, it's actually there's actually a smaller subset of DHS sites that actually overlap an HMR. And part of the reason for this has to do with the CpG density. So there's two categories of DHS sites. One where you see they're mostly methylated, and this is largely a group of DHS sites that have very low CpG density. And then the other fraction of DHS sites seems to be bimodal, and that's the green line. It's where you see that there are two types of DHS sites, ones that are highly methylated and ones that are not methylated. So this kind of indicates that perhaps the hypomethylation of these DHS sites represents an additional layer of information as to the activity of these potential regulatory or putative regulatory elements. So we wanted to characterize the intergenic HMRs a little more based on publicly available sets of chromatin state data. So again, as I mentioned, we used lymphoblastoid data as a proxy for B cells from ENCODE. And they also have, ENCODE includes H1, so they've done all of these experiments on H1 cells. So we have both the methylation state data and the chromatin state data for H1. So here I'm showing you a heat map of H1 cells and all of the different histone marks and ChIP-seq data that we looked at, including DNase hypersensitivity or chromatin accessibility measurements. On the top, what I'm showing you is whether or not every line, so this is sort of a uniquely represented heat map because we flipped it on its side. It's easier to see the different clusters of HMRs this way, I think. So every line, vertical line represents an HMR. And on top, I'm showing you whether or not they're shared with B cells or with lymphoblastoid cells, whether or not they're intergenic, so these are both intergenic and intronic HMRs that we've included here, whether or not they overlap a CpG island, so this gives you an indication of the CpG density at these sites, and whether or not, so there's an existing HMM model for calling chromatin state data. It's called ChromeHMM, and it's the one that was developed. At the Broad, and they basically categorized all of the regulatory elements that were identified by this data as according to whether or not they would consider it to be promoter or enhancer. So we're looking at whether or not our predictions overlap their predictions for these two categories. And so on the basis of this hierarchical clustering, we've identified 4 large groups. I mean, you can certainly see that there's substructure within each group, and it can certainly be further categorized according to that structure. But you can see generally that there are 4 big groups, and we've defined these groups as enhancer-like, promoter-like, bivalent, and CTCF. So the CTCF is the obvious one. This is the one where you don't see very much activity up here. You see really strong CTCF binding. In the bivalent category, these are the ones where you see active marks, both active and repressive marks. And this is the one that's probably the most important for this category, H3K27 trimethyl. That's very strong binding there. And you have active marks at the same time. That's why we call them bivalent. Like H3K4 dimethyl is a mark of an active state. But there isn't any H3K acetylated H3K27, and that's probably a mark that's associated with the most active state. So then we have a promoter-like state where you see very strong H3K4 trimethyl, and that's evident both in the promoter-like state and the bivalent state, less so in the enhancer-like state. So in the enhancer-like state, this is what I wanted to point out most of all, is that of all of the lines you see here, these are the least shared, or the most cell type specific. They also are the least likely to overlap an annotated CpG island. And it correlates very well with other predictions of this enhancer-like state. So we've based this entirely on our HMR calls and how they overlap with this chromatin state data. This IHMR classification is also supported by transcription factor binding. So the transcription factors in H1 cells that you would expect to be most associated with these different regions, such as this SOS12, the polycomb protein that's associated with this bivalent state. P300 is almost always located active promoters and enhancers. OCT4, an enhancer transcription factor for the pluripotent state, as well as NANOG, also enriched at enhancer. So we're encouraged that our classification strategy is fairly robust. So another thing I mentioned earlier has to do with the coordinated changes. So this was your question about how HMRs sort of interact with PHMRs, promoter HMRs. I'll say promoter just to be clear. So I mentioned that we see this spreading of methylation at cell type specific promoters. And that's kind of what this is showing here. If we look at differential methylation between H1 cells and B cells across B cell HMRs, we see that there's an expansion of hypomethylation in H1. So this is expanded H1 PHMR. Every line represents a promoter HMR in H1. And we're looking at the ratio of methylation in H1 to B cell. And we see that there's this region that's very shared in methylation state and a region that expands in hypomethylation in H1. And this correlates very well with the pattern that we would see in similar histone plots. So this is also the same, except we're not looking at a ratio here. We're just looking at H3K4 dimethyl enrichment.
So I have a question just to clarify, because you mentioned you're using the 2 kb upstream of the start site as the promoter. So 0 to 100 corresponds to minus 2,000 to 0?
Percent link. So yeah, it's actually is the only way we could normalize No. It's a percent length, yeah. Exactly. It was the only way we could, because there's so different lengths, and it kind of diffuses the signal on the outside of the HMR, so that's the only way we could normalize for length.
But it's always in the downstream to
yes.
into downstream direction.
Right. So we always plot it in direction that in the gene transcriptional direction. No.
So what's the length again? What is the length of
I'm sorry. What's
the
the
length of the distance between the HMR and the stars there? What is the length?
So it's not necessarily aligned according to the TSS, but rather I guess kind of it is. It's taking all of the regions across the TSS and aligning the HMRs according. I mean, they don't really. They don't. The TSS is almost always in the center of the HMR that crosses the TSS, but we actually only look at the one set of from one cell type. So one cell type, one set of HMRs, and then the corresponding methylation state across those HMRs for each line. And it's measured according to the percent length of the HMR centered on the TSS, I guess is the best way to say it.
So red means more highly methylated, right? So but that's relative to the methylation level of the HMR, or is it relative to the average methylation level of the genome?
It's relative to so this is it's a ratio. It's methylation at, I mean, it's sort of smoothed it's smoothed or bend across the region, right? But the measurement here is a ratio of the H1 methylation state within that window to the B cell methylation state within that window. Are we okay? Okay. So we wanted to understand whether or not changes at promoter HMRs are correlated with changes at IHMRs. So our hypothesis is that perhaps the presence or the appearance of an IHMR in a cell type specific context can be correlated with expansion of the promoter HMR and expression of a higher expression level of the gene, of the target gene. Now, I should mention here a specific caveat in this is that we're making one assumption, and the assumption is that the target of the enhancer is always the nearest gene, which isn't always the case. It's very often the case, right? But it's not always the case. Many times an enhancer can impart its activity on a gene that's much farther away, up to 100 kb away. And the only way to truly know what the interacting partner is for the enhancer is to do something like chromosome conformation capture, 4C or something like that, 3C to identify, or 5C, whatever C you want to do to identify the interacting partner for the enhancer. So, I mean, functional data is definitely needed. But in the absence of having this data, we were able to draw some information from the nearest neighbor idea. And that is that the red line is really the most important line here. And that's where we see that the expanded PHMR is closest to and is very closely located to an enhancer IHMR. So most of the promoter HMRs that become expanded in a specific cell type almost always have a partner enhancer intergenic HMR. And this is compared to an HMR that's not that is constant and never expands, which we assume to be sort of inactive or just not is expressed in a cell type specific way. And constant PHMR close to a CTCF HMR, for example. So a constant PHMR that is located close to an IHMR that is in the CTCF category is also it's not there's no correlation there. So and this is just basically reiterating the same thing, except here we're looking at the log-fold change in expression between B lymphocytes and H1 cells, and we're looking at distance to the closest IHMR. And at expanded PHMRs that are upregulated in their expression, they're much more likely to be close to an IHMR that appears in that cell type. Does this make sense? It's kind of confusing, maybe.
This is the change in expression?
Yes, this is a change in expression. Yes. A change in expression. And these numbers indicate the distance in KB in different bends.
Expanded is always of the more differentiated cell type relative to the stem cell, or because you could also shrink, right? If you start out with a larger one, right?
Yes, you can always shrink. In this case,
flip it so that it's always expanded, or
yeah,
leave out the ones that shrink.
I never. That's a good question, because I don't know that we don't look at it from that angle. It's always looking at the expanded, because honestly, like I mean, just to answer your question directly, I've not looked at shrinking HMRs. It could be, right? Yes, and they get actually shut down completely.
Okay, so they would not, right? You don't need them to be poised after that. You actually want to shut it down.
Right. Yep.
Just a question for the group: what's the median of the red curve? The median distance to the closest IHMR. We talked about this is a cumulative distribution function that we talked about earlier this afternoon. So what's the median?
Almost zero.
Is that?
Almost zero.
Well, the median is where half of the cases are below and half below. So when the curve hits 0.5, which is 50% of the cases, it will go up to 100% actually. So you can read it where 0.5 y equals 0.5 crosses the line. You go down. That's the value of the median.
This is good.
You said that the expansion seemed to correlate with the polymerase in your earlier talk.
It's a possibility. I mean.
Isn't that just showing that it's true right there?
I mean, it's sort of indirect data, but yes, I mean, it certainly would support that idea. That the expansion is correlated with higher expression level, higher polymerase activity at the PHMR. But, well, not necessarily more importantly, but the idea that the presence of an IHMR can be prognostic of the activity of a nearby gene is also very, very interesting. Because the changes, if you want to use methylation state as a prediction or something that gives you sort of a prognosis of what the gene is actually doing, it doesn't do it's so subtle. The changes can be very subtle at the promoter. So what I find to be the most exciting thing about this is that actually the presence or the appearance of a nearby enhancer intergenic HMR might be also prognostic of nearby gene activity.
Is there some level of clustering of expanded PHMRs in the genome? Do you see them kind of next to each other on the genome?
No, I think they're pretty randomly distributed, but and it depends on. I mean, they seem to be very cell type specific genes. At expanded PHMRs, you find significant if you only took expanded PHMRs and did some kind of Go enrichment, for example, you would find that those genes are highly enriched for specific cell type function. So
how do you interpret the correlation between the distance to the closest HMR and the changes in gene expression?
how do you interpret?
The correlation between the distance and the change.
How do you correlate? I mean, here, because this isn't a direct correlation that I'm showing. It's mostly looking at, well, exactly the log fold change at each distance from the gene, but it's not actually showing a direct correlation. So it's mostly inferred from this plot. I mean, I'm sure we could figure out a way to do it.
It's kind of striking that in the left half, the ones that are furthest away, right? I assume that it's 100 kb, right?
Mm-hmm.
The axis, it's a time point in the kb on the upper panel.
Right.
But you know, the genes that you shut down, it seems to be more at the level of maybe a locus control region or something, right? And that's more of a one gene level. If you know, it's striking that one is the furthest away are the ones that have the biggest effect, right? And then for the expanded ones, the ones that get activated, it's the ones that are closest to the DSS.
Right. I mean, well, and you mean, so I'm not sure. I'm not sure. Can you say it again?
Yeah, sure. So it seems, I mean, you have these 4 different categories of
right.
HMRs. Right?
They're different categories of distance. So they're different categories of distance from the nearest IHMR. And stratified according to whether or not the PHMR actually expands or not.
Right. But then the ones that are constant, right?
Mm-hmm.
The subset of those that has
I see.
IHR is farthest away.
Yeah.
Those show the biggest change in expression.
Right.
Which is kind of striking. You would think it kind of drops off with distance from the start site.
Right.
It's the opposite.
Yeah. I mean, they actually, it's. Yeah, I see what you mean.
Unless that means it's the next gene, right?
It could be, and this is, yeah, it's true.
It's striking.
Yeah. And it also may mean that, I mean, there's some regulation without expansion as well. So, but there's a lot going on in this. Okay. So I just showed you that we find coordinated changes both at promoters and at intergenic regulatory putative intergenic regulatory sites. So if we look at the if we just take H1 embryonic stem cells, we found that there's actually a subset of genes that have expanded promoter HMRs, which you would expect they would be upregulated, but they're actually silent. So they have, it's very interesting, they you know, there's something happening with the chromatin state at the promoter. It's getting expanded, but they're not expressed, they're not becoming upregulated, nothing happens. They're silent. And what we found is that there's a strong enrichment for the chromatin mark H3K27 trimethyl that's associated with silent states. And so it seems like these expanded promoter HMRs in H1 are in their bivalent state. And if you remember, I told you that we found many IHMRs and intergenic HMRs that are also bivalent. And if we look at the correlation between the H3K27 trimethyl enrichment at a PHMR and H3K27 trimethyl enrichment at the IHMR, we see that and we stratify them based on whether or not the PHMR and the IHMR are both bivalent. We find that the ones that we called bivalent are highly compatible with the promoter HMRs that are also bivalent. So there seems to be, even at this level, a coordinated change between the promoter and the upstream or downstream putative regulatory site. They're both being regulated in the same manner. So here's a nice example of, on the bottom, I'm showing you HMRs in H1 or B cells. And you can see a lot of different things going on here. On this side, we have a B cell specific HMR. It doesn't exist in H1. It has all the marks of activity, of chromatin activity, whereas those marks are absent in H1. On this side, we have an example of one that is actually a cluster of HMRs that are shared in both cells, but they're silent in H1. So there's strong enrichment for H3K27 trimethyl, and some enrichment for other marks, which makes this sort of a bivalent state. Whereas in the B cells, this locus becomes active, meaning you lose H3K27 trimethyl and you gain acetylated H3K27. So that's an example of changes that can happen between the stem cell and the differentiated cell. And here's one that's completely shared in all aspects between the two. So the last thing that I want to point out with this slide is that we see a lot of transcription along these areas. So I mentioned before that we see transcripts that are being produced from intergenic HMRs. And they don't seem to be associated with any annotated transcript or annotated gene or link RNA. They seem to be just completely independent of anything. So we wanted to further characterize this transcriptional activity at IHMRs. And so we wanted to compare transcription at IHMRs compared to mRNAs, link RNAs, also the different types of transcripts that are generated depending on the way we've classified them. And the take-home message is that different classes of IHMRs actually generate distinct types of RNA transcript, except for CTCF sites, which don't really, they're very quiet. But you can see that enhancer-like IHMRs and bivalent IHMRs, they're much more locally expressed. They're much more likely to be nuclear and less polyadenylated compared to mRNA transcripts. If you look at promoter prediction features of these transcripts, they're much less prominent in these categories than mRNAs and link RNAs, for example. So they're very distinct from link RNAs, which are also abundant in intergenic regions. And this slide is showing you that the transcription seems to be related to activity. And the way we've interpreted this is by looking at the methylation state at the same IHMRs in other blood cell types. So if you look at the methylation, if you look at B cell specific IHMRs, and then you look at the methylation difference at those sites to other cell types, you can see that there's much more, it's much different in H1, so as we expect at B cell specific IHMRs, the methylation state is much higher. And it doesn't matter whether or not we've considered the existence of an eRNA. If you look at other B cell types compared to other blood cell types compared to B cell, you can see that there's not, in many cases, there's at least where there's no eRNA in the B cells, there's not a lot of difference between methylation states between these cell types. However, if you look at the ones that are really B cell specific, so there is a slight difference between the methylation states at HSPCs and neutrophils. You can see that eRNAs really define that group. Where we have more eRNAs coming from these sites, they're much more likely to be more hypomethylated in B cells and what we consider potentially to be more active as a potential enhancer. And this is basically showing the same thing, that the fold enrichment at B cell HMRs of all of these different marks, including polymerase, is always much higher in B cell HMRs that are also producing eRNAs. And the reason why we call them eRNAs, we didn't coin this phrase. It was already coined way before us. There have been other groups that have noticed that enhancers can produce RNAs. So we're actually assuming that many of these are putative enhancers, and that's why we call them eRNAs. Okay, so because of this this observation, we wanted to understand what the characteristics of the if there are any sequence features that coincide with the transcription start sites of these eRNAs. And so basically all of these plots are lined up on the TSS. So we looked at cage data, which gives you a sort of a defined 5' end for these eRNAs. And so we lined everything up on the TSS as defined by cage. And then we looked at different sequence features. So you can see the cage. This is basically looking at the cage peaks. This is looking at histone enrichment aligned to the TSS. This plot is showing, so this is showing GC skew. So this is CpG density, and this one is showing the bias in C's versus G's across the TSS. It's actually a phenomenon that's been noticed at the TSSs of many promoters, and it's believed to be involved in R-loop formation. So it's suggestive of RNA polymerase activity. And the ART score, which is basically a tool for predicting promoter-specific elements in sequences. And it's sort of trained on mostly real promoters and not necessarily these types of regions, but you do see a slight peak, even though the score isn't very high. We also found an enrichment at the TSS for TATA binding proteins, both Pol2 and TATA binding proteins and other members of the transcription factor transcription complex. And we actually found that, because of this, we were interested in looking to see whether or not there's an enrichment for a TATA motif, which is often very difficult to find because it gets very degenerate, and a lot of people have found that it's very difficult to find a real TATA motif at most promoters because of this. But we actually looked for strict TATA motifs, and we found a pretty good enrichment of TATA motifs that correspond to the TSS of the eRNAs that are produced from these sites. So this indicates that there's really, and we also noticed that at the very site or position of the TATA, an increased level of conservation. So it would make sense. So this plot is just showing where we find the fraction. So this would be the position of the TATA and the fraction of the region covered by cage tags, and it's actually because the TATA box is usually a fixed distance from the TSS, so it looks very real. And we also asked whether, in this case, for every category that we defined of IHMRs, we asked what percentage of IHMRs have a TATA. Yes?
So it seems in some of the literature say that the transcription of the constantly bidirectional. So do you going to see enrichment both?
Yeah, so in this case, we've actually looked at the direction of the, so actually there is a lot, I mean, many of the transcripts that are produced from the eRNAs are directional, and they're unidirectional and often in similar direction to nearest genes, I believe. In this case, I mean, we always look at unidirection, so the most likely direction of that are shown by the cage tags. So, I mean, this one doesn't really show you very well, but we often see. I mean, this is basically showing that it's actually quite. I mean, most of the it's an aggregate plot, but it's showing that most of the transcripts that are produced from these regions are directional.
How do they transcribe and answers characterized by bidirectional transcription? Like usually
Sorry?
cage text distribution on transcribed and answers is bidirectional. So you typically see signal. I think.
Yeah, I mean, you see, you can see. I mean, yes, it's true that you can see bidirectional signal from these cage tags, but usually, I mean, it's at enhancers, yes.
And then usually then at real transcription start sites, you see the more directional.
Right, which is what this is showing. I mean, you can see some signal in both directions, but for the most part, it seems to be biased on, yeah, yes. Right, so as I was explaining, percent of IHMRs with a TATA. So if we look at our different categories that we've classified for the IHMRs, the enhancers are the ones that really have the enrichment for the TATA box, and it's much more enriched at enhancer like IHMRs that have cage tags. Okay, this is my next to last slide. So this begs the question of whether or not, as we discussed earlier, there is a potentially causal role for transcription in the establishment of hypomethylation.
Can you go to the previous slide?
Yeah.
Pole 2 as tags and pole 2.
I'm sorry?
The previous slide.
Yep.
So which form of pole 2 are you referring to?
Which form of pole 2? I can tell you after. I don't know. It's I'm not sure exactly which one we looked at here, but it's based on this series of ChIP-seq that was done by ENCODE. So there's several different forms of the antibodies that were used. We can look it up. I don't know right off the bat, but we can look it up. Right, so just looking again at going back to the question of DNA's hypersensitivity, we can see that the ones that actually overlap hypomethylated regions are more likely to be conserved. The DHSs that are hypomethylated are also more likely to be expressed. So this is looking at cage versus the cage in RPKM versus methylation level. So the lower the methylation, the higher that's the likelihood that the DHS will also produce some kind of transcript. And then looking at whether or not the DHS has a TATA box. The ones that have a lower methylation level also seem to be more likely to have this TATA, conserved TATA motif. So just to summarize the points that I tried to describe to you, patterns of methylation distinguish developmental lineages, and in fact, these intergenic patterns are the most cell type discriminatory. We were able to integrate numerous genome scale data sets to facilitate the clustering of these non-promoter intergenic HMRs into functional groups with very discernible regulatory features. And permissive DHS sites seem to already be set up in embryonic stages, but even though they're hypersensitive, they can still be methylated. And we'll have one more thing to show you after this. And transcription seems to be a hallmark of active IHMRs, which we would describe as putative enhancer elements. So this is our working model right now for trying to put together everything I've showed you. So we're looking at embryonic stem cells, then the intermediate step, the adult somatic stem cell, and then the terminally differentiated B cell. And what I'm trying to show you is that in many cases, so at the CTCF IHMR, for example, you have constitutive DNase hypersensitivity and constitutive hypomethylation that's shared between all cell types. But at other IHMRs, such as the enhancer IHMR, the DHS, the hypersensitivity, is already pre-established in the stem cell, and then it progressively becomes hypomethylated as you differentiate to the most differentiated cell type. This demethylation or de novo demethylation of this site is associated with active histone marks and the transcription of eRNAs, and it seems to be coordinated with the expansion of promoter HMRs and increased expression of those associated genes. So with that, I want to acknowledge everyone who has helped me in this work, everyone in the lab, Greg, my boss, Andrew Smith, and Felix, who have done a great deal of work on this, and hopefully we can talk a lot more about this after. Thanks. Mm-hmm.
Is it possible that they're just competing with the maintenance methylation?
There's a lot of possibilities, and so I didn't want to be kind of unilateral on that statement, because I think there are examples in the literature, and it hasn't been very well characterized either. I think this is still up for debate, but for the examples that there are, I think that some transcription factors can bind with varying levels of affinity to methylated DNA. But then there are others that actually really prefer an unmethylated substrate. So I don't think that there's obviously never a black and white scenario.
I was wondering if in the B cells, maybe there are more of those particular transcription factors that have binding
Compete.
to the HMRs that you find.
Right. There's certainly a very strong enrichment. I mean, we looked specifically at the transcription factors that are available, the data that's available, so already the experimental data for actual binding of the ChIP-seq data. And so, I mean, that certainly seems that among the ones that are included in those data sets that are very lymphoid specific, that there's a very strong enrichment for multiple transcription factors. Sometimes you have numerous sites for the same transcription factor, and then obviously multiple different lymphoid-specific factors that are all piling up together there.
So how do these cells know how to put these marks? Does the profile track specific DNA methyltransferases, or the profile you see is specific to certain transcription factors that make these marks?
How to answer this? I don't know. I mean, I think the profile that we see, I mean, I think there's basically two things at work. Because you have the DNA methyltransferases that are sort of responsible for continually methylating DNA. And as you mentioned, there must be some kind of interference with that machinery that prevents DNA from becoming methylated. Because I think DNMT is pretty promiscuous. I mean, it likes to do its job, but there are kind of interfering factors that can prevent that. But at the same time, even though we haven't necessarily identified an exact enzyme that does demethylation, it's believed that there are both active and passive mechanisms for demethylation of DNA. And I actually didn't discuss that. I mean, here, what I showed you is that in differentiated cells, you have very cell type specific IHMRs, which implies that there's some kind of demethylation of those regions, because they don't exist in the stem cell. But we don't know why, and we don't know what it is.
Nucleosomes. Is there anything known what's going on with the nucleosomes? Changes that you can tell us about, have you compared nucleosome patterns with these profiles?
Yes.
Is this just another thing that correlates with DNA swan, and you get nucleosome depletion correlated with that, or could it be something?
I can show you. I can actually show this if I can find it. This is freaking me out. This is so like, where do I find my Dropbox?
You can draw it on the board.
Yeah, I mean, if you compare nucleosome patterns across these HMRs that are cell type specific, it looks like a very, very typical nucleosome plot where you see something like, I mean, you see very strong depletion of nucleosomes at the cell type specific places, and and if you look at the same thing, if you look at the nucleosome pattern from one cell type at the specific HMRs of the other cell type, then it just looks like it really looks like something like that. It's nothing. It's very flat, just a little bump.
Did you get a chance to look at tumor cells? I mean, methylation is really messed up in cancer cells. Is that something that you're pursuing? Is that something that
Yes. We've been looking in the lung cancer cells, and what we find is, well, so there's actually two parallel phenomena that are happening, I think. One is that we see hypermethylation of promoters. So this is what many people have already reported, that you get hypermethylation of promoters, and then you get this widespread loss of methylation outside of genes. So really, really widespread. Seems to correlate with lamin-associated domains. But interestingly, if you look at cultured cells, they show very similar patterns. You see widespread loss of methylation in intergenic regions, and it's not necessarily a feature of only cancer cells. So if we can sort of separate these two things, I mean, certainly in cancer cells, the loss of methylation outside of genes is very dramatic, but it's also something that you can see in cultured cells too. So I think we have to be very careful to sort of distinguish those two things.
Yeah, what's happening to your HMRs in cancer cells? I mean, this hypomethylated epigenetic.
Right.
What's happening to them?
That's something we're looking into now. and I don't really know the answer. They're just so screwed up, and yeah. You had a question? So, for example, something that's been immortalized. It's like, yeah, I think. Well, so let's say cells with normal karyotypes that were otherwise completely normal before they were transformed or EBV immortalized, for example. There. I mean, I agree with you. There's certain. Even the cancer cells that are, I mean, if you take a direct tumor cell, tumor tissue, and look at methylation patterns versus the ones that have actually been cultured, they do look different. There definitely seems to be a cell culture effect.
I've worked on similar things in extra-embryonic lineages, and they've done studies where they derive them from a mouse, and they can follow the methylation change, like the feature capacities from culture, without any sort of transformation problem.
Yeah.
Transformation problem with EBV, and so just cells in culture to keep going, they have to.
Okay, well, if there's no more questions, let's thank Emily again.
Thank you.
And, see you in the other room in about 10 minutes.